Hello, my name is Amir Goldkorn. Thank you for the opportunity to tell you a little bit today about the USC Norris Liquid Biopsy Core and the way that we're facilitating a new biomarker and therapeutic discovery through some of the technologies that we offer. So the challenge with phenotyping advanced disease is that obtaining biopsies of primary tumors or metastases can be difficult and risky sometimes to patients and offers a limited amount of information. As an alternative, liquid biopsies can be drawn multiple times through peripheral blood draw and can be analyzed for various analytes such as circulating tumor cells, CTCs, cell-free nucleic acids, extracellular vesicles, and so forth. The difficulty is that circulating analytes are rare, needle in a haystack. In a standard tube of blood, you might have 40 billion red cells, 40 million white cells, and maybe only five or so circulating tumor cells. So our space is really bridging the new technologies that are becoming available over time and incorporating them and developing workflows to actually take them into the clinical setting where they can be used to answer biological and clinical questions. To do this, we've gathered a variety of instrumentation and technologies that enable us to enrich rare materials from the blood. We use this in a variety of ways. Here's an example of where we look at an established biomarker using liquid biopsy. So this is a large metastatic prostate cancer trial where in one of the arms, we're simply using the FDA cleared cell search platform to count cells. And what we showed was that men entering the trial who had no detectable CTCs had a ninefold chance of having a complete response to hormonal therapies and continuing to have that response at 2 years. In other work, we've been developing new workflows, for example, looking at expression profiling from circulating tumor cells, here using the Parsortix platform to enrich using a microfluidic approach, live CTCs. We incorporated this into a new workflow that allowed us to deplete away background cells and really go in 3 hours from patient to RNA, where we recover about a third of the CTCs, and all those background cells are depleted down to maybe about 100 cells. This allows us to go into an RNA-seq workflow and generate the type of volcano plots and pathway analysis graphics to look at various cancer-relevant gene expression pathways that now can be detected and looked at in patients with metastatic disease over time from the blood. Similarly, we're developing biomarker workflows using the cell-free DNA from the plasma in the liquid biopsy. Here's one example where we're looking at cell-free DNA methylation profiles. These are patients with bladder cancer receiving neoadjuvant chemotherapy, and we're able to separate using machine learning here based on the methylation profiles, the non-responders here in red versus responders to chemo, beginning to generate some classifiers that allow us to predict who's going to have complete or partial or no response to the chemotherapy. Ultimately, the vision is to combine this into what we call a multi-parametric liquid biopsy that allows us to look at all parts of the elephant, as it were, by generating these larger profiles of cell-free DNA, cell-free RNA, CTC, mutations, copy number variants, and looking at them again over time to see how these evolve. We're further streamlining this with workflows that allow us to get all the cells onto slides, stain, identify cells, recover cells of interest for sequencing, such as this workflow here, where we get the CTC counts, we stain for things like synaptophysin or ARV7, this is a prostate cancer, and then recover single cells for somatic mutational analysis and compare them to the analysis from the matched plasma from the same tube of blood. This type of workflow has actually been incorporated into a new phase 3 prospective trial where we're collecting blood over time and looking at these various analytics. We're actually also combining it with radiomic analysis and targeted sequencing. So in summary, liquid biopsy can be used to characterize tumors over time in clinical and translational settings. Our NCI designated core has capabilities that could be leveraged to identify the presence of clinically relevant cancer phenotypes, quantify expression of signaling and resistance pathways, and recover materials for next-gen sequencing for things like copy number variants, mutations, and even methylation. I've included our link here at the bottom for anyone who would like to learn more, and we'd be happy to talk with you. Thanks so much.